A very good day to everyone. Today in this session, we'll be mastering the immunoassays from CSIR DBT JRF point of view as well as from Gate Biotech point of view. So we will study about all the comprehensive principles, methodologies and applications of different types of immunoassays. Now, before we move into what an immunoassay is, we should first learn about different type of antibodies. Now, a monoclonal antibody is something which reacts with only a specific epitope given in any antigen. For example, if this entire blue is your say an antigen, so there are different types of epitopes which are present on the top. Some may be yellow, blue, orange and purple. So if you see, this particular antibody is specific towards this blue triangle only. It is not going and binding to the purple, orange or the yellow one. So this is known as your monoclonal antibody. Whereas on the other hand, your polyclonal antibody can recognize or it can bind to more than one type of epitopes on any given antigen. For example, this particular antibody can recognize your triangle blue antigen epitope, or your purple square, your orange square, or even the yellow circle. So they are known as polyclonal type of antibodies. So this antigen antibody interaction is very important not only in immunology, B cells or T cells, but for doing any kind of immunoassays. So the first kind of immunoassay what we will be studying is radioimmunoassay. In most of the immunoassays, what you will be finding is we are dealing with generally monoclonal antibodies, that is which are very specific towards one antigen and they can very specifically go and bind to that particular antigen. So this radioimmunoassay was first developed by Rosalind Yallo and Solomon Person in the year 1960s, and they first used it for detecting the level of insulin in a human blood sample. Radioimmunoassay is considered as a very sensitive and very specific assay. So you can detect any particular hormone, any particular serum or protein, drugs, whatever you want in a given sample. Here we use antigens to detect and quantitate any particular type of antigen or analyte, whichever is there in your sample. So even if your quantity of antigen is very low, even in picograms if it is, we can still analyze it. Now, how does this principle actually work? We have some primary antibodies which are very specific towards a given antigen, so we will radio label them. So this radio labeled antigen or radioactive antigen goes and binds to most of the primary antibodies. Then you can add your sample. Now remember, the sample what you have added generally contains unlabeled antigen. So if you add, or suppose if your sample contains more number of unlabeled antigens, obviously they are going to go and replace this labeled antigen from the particular antibody. So few antibodies might have completely removed the radio labeled antigen, and few might have unlabeled and radio labeled attached both. Then all these primary antibodies which are attached with your antigen, specific antigen, whether it's radio labeled or unlabeled, they are attached or they are reacted with a secondary antibody which is again very very specific towards your primary antibody. So now when you pull this secondary antibody out, or when you just analyze this secondary antibody, you will see that few of the primary antibodies will be having radio labeled antigens attached with them and few will be having non radio labeled antigens attached with them. So now what happens, we just need to take and analyze it with any I, radio assay, whichever can read what all kinds of radioisotopes or radio antigens are present here. So you will get generally a decreasing graph. Decreasing graph means if your unlabeled antigens are more in number, so eventually as you increase the time, you will see that the concentration of the labeled antigen decreases. So if the radio labeled antigen decreases, then that means the unlabeled antigens have gone and occupied that primary antibody's binding site. So then we can infer that our sample contains more number of unlabeled antigen. If a person is having more number of antigen, that specific antigen, remember the radioactivity is going to decrease. So this is an inverse relationship. This is the principle of your radioimmunoassay. So this is just an example, a quick recap for us, that the serum of an uninfected person can be taken as a control, or they can have very low concentration of that particular radiolability. You have taken an infected serum also. So when you take infected serum, so the number of unlabeled antigen would be more and number of labeled antigen would be less. So let them bind with your primary antibody. Once they bind, few areas would be occupied by your unlabeled antigen and few areas will be occupied by your labeled antigen. Whereas in your control sample, where you are not putting any infected patient's sample, remember they are having only radio-labeled antigen present in the sample. So all the spaces are occupied by your radio-labeled antigen. Now you do the radioimmunoassay. So obviously the person who has infected with that particular antigen or who has this unlabeled antigen present will show a decreasing graph. That means this number of the number of unlabeled antigen in the infected serum has replaced your radio labeled antigen, and therefore the graph decreases. So more your graph decrease, that means more number of unlabeled antigen is present in the sample. So this was the principle of your radioimmunoassay. Second, we will be studying about ELISA. ELISA is known as enzyme-linked immunosorbent assay, very commonly used in most of the biotech labs, molecular biology labs, and immunological labs. ELISA stands for your enzyme-linked immunosorbent assay. This is a very common laboratory test whenever you want to detect or you want to quantify any specific antigen or any specific protein, hormone, any specific substance which is present in your given liquid sample. It uses very high specific interaction between antigens and antibody. So this was first described by Engvall and Perlman in the year 1971. It is commonly used to measure any kind of particular antibody present in the sample, if any antigen, any protein or any glycoprotein in any of the biological sample. ELISA was first used to diagnose your HIV infection, and now its application has increased far beyond just simply detecting HIV. You can do pregnancy test detection, or you can measure what kind of cytokine or soluble receptor is there in any cell supernatant. Other than that, you can detect any kind of infection, whether it's HIV or thyroid or insulin hormone. You can detect any hormonal level also. Now, how does this work? Here, you have a plate, or generally an ELISA reader plate or a microtiter plate. So you can use either antibody or antigen. So they, as we know, they will covalently bind to each other. So this reaction again becomes very specific. ELISA is again of four different types. You have direct ELISA, you have indirect ELISA, you have sandwich ELISA, and you have competitive ELISA. So we will see each of them in detail. Remember, the antibody which you are using, it is generally conjugated with an enzyme, and this enzyme has the tendency that the moment you add a substrate, this enzyme will convert it into a colored product. So you can find a colored product, or you can find a fluorescent labeled product, or you can find a chemiluminescent product also, which is very easy to just take the reading under any spectroscopy. Now, it is generally carried out in a 96 well ELISA titer plate, or even more 384 well plated ELISA plate reader you can do. The advantage is you can do multiple samplings here within a single experiment itself. Most commonly used enzymes which we use for conjugating it with the primary antibody or with the antibody is your HRP, that is horseradish peroxidase, or alkaline phosphatase, or even beta galactosidase. So let us quickly see about the direct ELISA. If you see, direct ELISA is quite simple, although it is less sensitive, but it is quite simple. The antigen here, it is mobilized first on the 96 well plate. Suppose we have a microtiter plate or a ELISA plate or having 96 well plate. So we will first coat our desired antigen on the surface. Then you add a primary antibody. Now remember, this primary antibody is very specific towards this antigen. So your primary antibody is conjugated with an enzyme. Either of these three enzymes, you can just conjugate it. The moment you add a substrate, the substrate gets converted by the enzyme into a colored product. It could be a colorful product, it could be a fluorescent product, or it could be a chemiluminescent product. This immediately you can measure it under any spectroscopy technique. You can check the absorbance at any particular given wavelength as per your experiment. So the more color or the more reading that you get, that means your sample has more number of antibodies or more number of antigen present in it. That is direct ELISA. However, direct ELISA is considered little less sensitive since we have only one check here. We are using only primary antibody. The second type is your indirect ELISA. Now, what happens in your indirect ELISA? Here also you are coating the well with an antigen. You wash off if anything excess or extra is there, which you do not want to hamper your experiment. Then you add the primary antibody, which is very, very specific towards your antigen that you have coated. You wash off anything excess, and then we add a secondary antibody which is conjugated with an enzyme. Now remember, the secondary antibody which is conjugated with an enzyme is very, very specific towards your primary antibody, and in turn your primary antibody is very specific towards the antigen. So the moment you add a substrate, it gets acted upon by the enzyme and gets converted into a colored product. You just have to take the OD. The more the color, the more is your sample present, or the more is the antigen or antibody present in your sample. The third is known as sandwich ELISA. Sandwich ELISA is even more specific and sensitive than your indirect ELISA. Here we coat the wells with an antibody, you wash off anything excess, then you add your antigen. Now your antigen goes and binds to the primary antibody very specifically. Again, wash off anything which is excess, not required. Then you add your secondary antibody, which is again conjugated with an enzyme. The moment you add a substrate, it gets converted into a colored product. This is called as sandwich. Why? Because your antigen is sandwiched between the primary antibody and the secondary antibody, and hence the name is as sandwich ELISA. This is also very sensitive and very efficient, even more than your indirect ELISA. The fourth is known as your competitive ELISA. Now in competitive ELISA, remember, there will be few antibodies which have already, or which onto which you will already add the antigen, and few of the antibodies will be naked. So both these are very specific towards one common antigen. The moment you add them, you have a microtiter plate which is coated with your antigen, the same antigen which is bound here. So the moment you add these two antibodies inside, remember, the one which is already incubated or bound with an antigen cannot go and react with these red dots or with these red antigen, because their site is already filled. The one which is empty, this only will go and bind to the antigen which is coated on the well. You wash off anything excess. So this one, the one which was pre-incubated with an antibody, it just gets washed off. The moment you add a secondary antibody conjugated with an enzyme, you add substrate, it gets changed into a colored substance. Now, the moment you get a colored product, remember, only the ones which are not, or which were not initially pre-incubated with the antibody, only they can go and bind. So therefore, here is a competition between your bound antibodies and the unbound antibodies. If you get a darker reading, that means a lot of unbound antibodies is present in the patient sample. If you get a less color, so that means less number of unbound antibodies were present in the sample. So these were the four type of ELISA test which we have done. The next type of immunoassay is immunofluorescence assay. Now here what we do is, this was first described in the year 1944 by Albert Cones, and here also they use the same principle of antigen antibody interaction. The only thing is, your antibodies could be labeled with a molecule that has fluorescence property. Now, instead of conjugating it with any enzyme that can convert the substrate into a colorful product, you are directly using a fluorophore, that is a fluorescence compound. Now, this fluorophore molecule will absorb exactly one wavelength of light. It gets excited. Now, once it gets excited, the electrons will move from the ground state to the higher energy orbital. Then, after some time, they will come back to the ground state. So in this, while they were coming down in the ground state, they will release or all that energy, what we call it as the emitted fluorescence light. This gets detected by your microscope, and this is the basic principle how it works. There are two types of immunofluorescence assay: direct, in which you have an antigen and you just add the antibody which is directly attached to a fluorophore. So the moment an interaction is here, you excite this particular complex at a particular wavelength and it will just fluorophore. The second type is your indirect immunofluorescence assay, in which you have an antigen, you react it with a primary antibody which does not have a fluorophore, and you have a secondary antibody again which is very very specific towards your primary antibody. Your secondary antibody has fluorophore attached. So now when you hit it with light, this entire complex is going to glow. So your indirect immunofluorescence assay is little more sensitive and little more accurate in giving the reading. These are the different type of fluorophores that we use, either PE or fluorescein or PERP CP or Texas red or rhodamine. In most of the experiments, you will be using fluorescein or rhodamine since they absorb or they fluoresce at a different wavelength. Your fluorescein is mostly going to absorb blue light and it emits a yellow greenish light, whereas your rhodamine will absorb your yellow green range and it will emit a deep red range fluorophore substance, which can be easily detected. The next technique is your immunohistochemistry. Now, this is also most commonly used application in your immunostaining. Here, what we do is again the same principle. Instead of using one particular cell or one particular antigen, you have selected an entire tissue. You prepare a very thin section of the tissue, you fix the tissue, you prepare a very thin section of the tissue, and then you go for staining. The antigen or the protein which you want to target, depending upon whether it's on the cell surface or within the cell surface, you can permealize it, but that particular protein becomes your antigen. So you will construct a primary antibody which is very very specific towards this antigen. The moment you add the primary antibody, it goes and binds to your desired protein which is present in your fixed cell. Then you add a secondary antibody. Here your secondary antibody is again conjugated with an enzyme. So the moment you add a substrate, the same enzymes which we used in the previous experiments, the moment you add substrate, it gets converted into a colored product. So if you get a colored product, so that means your particular desired protein or antigen is present only on those cells in this entire tissue. You can just simply see it under any fluorescence microscope, or you can just simply see it by igniting. You can use an enzyme or you can use a fluorophore also. It depends upon what kind of experimentation you are doing. You just have to visualize it by using proper means. If you are using any color changing substance, for example the enzyme which we saw, enzyme changes your colored uncolored substrate into a colored product, that is known as your chromogenic immunohistochemistry. Chromogenic means change in color. So we use enzymes such as peroxidase which changes your uncolored substrate into a colored product. The second type of immunohistochemistry is immunofluorescence. Now here I am not tagging my antibody with any kind of enzyme as you see here. Here I am tagging my antibody with a fluorophore, which could be a fluorescein or a rhodamine, depending upon which color you want or which experiment you are doing. So you just have to hit it with a particular wavelength of light and it fluoresces. So here you are not changing the color, you are just observing the fluorescence change. So there are two types of immunohistochemistry: chromogenic for color and immunofluorescence for fluorescing substrate. So this is a detailed methodology how we do it. First we prepare the tissue. We first have to fix the tissue. You collect the tissue, you fix it, and you prepare very thin sections of that tissue. Once the tissue gets fixed, you can dip it in different kinds of alcoholic washes so that there is less amount of water content that does not hamper your experiment. Once your sample is prepared, you go ahead with sample labeling. So the protein which you want to identify, add primary antibody against that particular protein. Then you add a secondary antibody which is conjugated with an enzyme. Color changes your uncolored substrate into a colored product that becomes your chromogenic, or you can use a fluorescence for your secondary antibody. You have that antigen, add primary antibody specific for your substrate, specific for your antigen. You add a secondary antibody having your fluorophore and just ignite it or excite it with the desired wavelength of light. So you can observe the fluorescence also, which is immunofluorescence. So these are some of the beautiful images which you can see. For example, here this particular antibody having a fluorescent molecule attached, it goes and binds to the red type of receptors on the cell. The moment you excite it, it will emit different kinds of green light. So all the cells which you can see greenish, they are your targeted cell. This is the principle of your immunohistochemistry, where we have also used immunofluorescence. That is, I am using a fluorescent molecule here. I am not using a substrate, or I am not using an enzyme which converts a colorless substrate into a colorful product. So you can see you can use more than one type of fluorescence. For example, the blue ones, they are the nucleus. The green ones, they are your actin filaments. The red are different types of proteins which are present on your in your cytoplasm. This is again a beautiful picture of immunofluorescence microscopy. If you see here, antibody is conjugated with fluorescent dye. So you can use more than one dye. You can use red or green and yellowish or and bluish also. So they are going and binding to their respective sites. These are primary antibodies conjugated with your fluorescent dye. They will identify the protein as per lock and key hypothesis. So they go and they bind it, and then you can just simply identify it under any fluorescence microscope. For example, this bluish region, all the antibodies which are specific towards the nucleus, proteins of the nucleus, will go and bind here. You can see them blue under the microscope. Similarly, the red ones could be for the cytoplasm. The green and this yellow one, they could be for different targeted proteins. The clinical research application of all these immunoassays is they are generally used for your diagnostic and oncological processes. You can do any kind of critical clinical diagnosis. Any disease, any antigen can be detected, whether it's HIV or hepatitis or dengue or even COVID SARS virus. So you just have to be sure about the antigen and antibody reaction here. The second is in your endocrinology. So you can quantify any vital hormone, whether it's getting upregulated or downregulated, such as your TSH, your T3, T4, insulin, human chronic gonadotrophin, so, or the basic of pregnancy test. So you can go ahead with endocrinology also. Oncology markers, any kind of marker which gets presented on your cancer cell. For example, PSA in prostate cancer cell or CA125 in the ovarian cancer cell. So the antibody goes and binds to these particular targets, ovarian oncology markers, and if you see a positive signal under fluorescence or under chromogenic kind of techniques, so you can get assured that these particular cells are having oncology properties. Talking about pharmacology and research, they are used for developing therapeutic drug or in general therapeutic drug monitoring. So many kind of narrow index drugs such as your tacrolimus or your dioxins, they you can ensure their efficacy and prevent any kind of toxicity. You can see that they are not going in cross binding with any other cell. And also in normal Western blotting, we use such kind of techniques, a standard validation process for identifying the presence or absence of any particular given protein. So for these purposes also we use immunoassays. So I hope the session would have been beneficial and quite interesting for you. If you have any doubt, feel free to reach our support chat support system. Thank you so much.